Hello, a very warm welcome to the Pharma session of Waters Asia Pacific LC Symposium 2021, Reducing OOS Results in the Pharma Lab. My name is Terence and I'm currently working as the APAC Pharmaceutical Market Development Manager in Waters. I'll be your host for today's event. As we know, out-of-spec results may arise from poor analytical method performance, and this can impact time to market, revenue, and reputation. In this session, our speakers will explore the different tools and best practices on how to reduce out-of-spec results by improving upon method performance to drive data quality, be it in routine use or when transferring your methods to different systems or geographies. Before we begin, I would like to draw your attention to two main housekeeping items. Firstly, on your screen, you can see the main presentation screen. If you hover your cursor to the CC button, you may select the appropriate language subtitles according to your preference. Secondly, I would like to invite you to join us on Slido for Q&As and polls. You can do so by directly clicking on the link provided to you. Join us on Slido on the top left-hand corner next to the main presentation screen, or alternatively using the event codes or QR codes provided to you. On Slido, you may type your question in English or local languages such as Korean or Japanese. Best of all, you can simply like other questions without asking one yourself. Last but not least, please take a few minutes to complete the survey at the end of today's live event. Now I'd like to pass over to our first speaker for today, Charlotte, who will introduce the current Waters liquid chromatography portfolio.
Hello, and thank you for joining us today. My name is Charlotte Yu, and I am a Separation Business Development Manager at Waters Asia Pacific. As many of you here already know, Waters has launched several new products in recent years with innovative technology. To start today's session of the LC Symposium, I'd like to start by giving you a short overview of the current Waters S portfolio. As a scientist, we have questions every day. Do my samples contain toxic substances? Is our product safe? Is my data accurate? Our goal is to help you, our customers, confidently make critical decisions to move your business forward. From fundamental research to method development to routine testing, our integrated, robust offering of instruments, chemistries, informatics, and support addresses your research complexity. Our chromatography portfolio provides you with abilities. First one is usability. Our platforms support a consistent user experience, whether you are using an LC with optical detection for routine analysis or an LC with mass spectrometry for trace level analysis. Scalability. Our portfolio allows you to scale to meet your needs regardless of chromatography platform, column, or solvent. For many applications, LC configurations are paired with consumables and informatics, minimizing the time being spent from sample to result. Transferability. Our long-standing LC methods will run from instrument to instrument and lab to lab, producing the exact same data. As you develop or transfer your methods, you can rely on Waters to keep them fit for purpose as they move from development to routine use. Dependability. Our application scientists and global technical support teams answer your questions, help develop methods, and problem solve every step of the way. We are here to maximize your research and the innovations that result. There are many examples of the innovations in LC that we have brought to market in our history as a leader in separation science. The Alliance space has been the backbone of many labs for over 20 years. But there may be no better example of how Waters has enabled scientists to gain competitive advantage than the advent of the Equity UPSC in 2004. Since 2004, each addition to the Equity family has been designed to allow a broader range of scientists to benefit from this game-changing family of products without compromise what they do. And with this industry following Waters' lead, the face of chromatography has changed. We have also added new specialty separations technologies such as convergence chromatography with UPS squared, polymer characterization with the Equity APC system, and process monitoring with Petrol. Finally, we introduced several products in recent years to meet customers' needs and to solve another challenge. The Waters' portfolio can be broken down in many different ways, so I would like to introduce our portfolio in 5 overlapping groups. The first one is analytical LC systems. These systems allow you to conduct your routine analysis with confidence. This group includes the original Equity UPLC, the H-Class Plus, the Achilles PLC, and the Equity Premium. These products provide the flexibility for easy method development, reliable method transfer, or uncomplicated routine analysis. Next one is LC for MS. High-performance LC technology delivered in optimized configurations that power up the sensitivity, reproducibility, and robustness of your MS analysis. Here I would include systems such as the Equity I-Class Plus, the Equity M-Class, and new Equity Premium system. Next, we have system solutions. These fit-for-purpose solutions, like the Petrol system or the Equity APC system, were designed to address application-specific workflow requirements, complex matrix analysis, or production environment operating constraints. We also have simple separations. These systems are ideal for those new to separation science. These core technologies, such as the Alliance HPLC or the BridgeQS HPLC, offer affordable, highly reliable, low complexity systems for routine analysis. And finally, purification systems. Preparative scale HPLC plays a critical role in applications where compounds must be synthesized, identified, isolated, purified, and characterized. These systems enable substantial increases in throughput with an added dose of system robustness. But the chromatography separation itself is only one piece of the puzzle. We also need to collect, analyze, report, and secure our data to be able to make critical decisions with confidence. Our informatics solutions capture, manage, integrate, and analyze your LC and MS data, improving discovery and manufacturing, quality and integrity, and reducing time to market. Easily and seamlessly conduct scientific searches, integrate networks, manage compliance, and streamline operations. You invest significant capital in your LC system. Why not use the very best consumables? Waters is committed to developing, producing, and manufacturing innovative and consistent products for solving your most difficult challenges. As you can see here, our enabling technologies meet your standards for performance, reproducibility, and quality, giving you confidence in your analysis. As I mentioned before, Waters has added 2 new systems to our portfolio in the last 9 months, the Achilles PLC and the Equity Premium. So lastly, let's take a look at the benefits of these products. The Arc HPLC was released in the summer of 2020. It is built on Waters' proven technology. Method transfer could be transferred between different labs or different vendor instruments or different separations platforms. The Arc HPLC is ideally designed to show equivalent separations between different platforms, not only for Waters instrument, but also other vendors' HPLC systems. We can use features such as Gradient Smart Start to facilitate this process, saving time, leading to more productivity and better asset utilization. If you want to improve your productivity and efficiency, this can be performed on an Arc HPLC as well because of its higher back pressure tolerance compared to the existing Waters HPLC. We can scale the method, moving to a 3.5 micron column, and all performance can be improved. And additional benefits include lower solvent purchase and disposal costs. As the next product, in February 2021, Waters redefined separation science once again with the introduction of the Acute Premier. This system solution integrates consumables, column chemistries, and the LC system to solve some of the most challenging separations in analytical chemistry with mass spec technology. What are the challenging separations today? Honor-light loss is one of the most troublesome challenges in chromatography that labs experience today. This problem manifests itself in many ways: poor sensitivity, poor reproducibility, poor recovery, or even missing analytes. MaxPeak high-performance surfaces technology, which we are applying to the LC system, the column, and the consumables used in the Premier solution, are designed to improve sensitivity, increase analyte recovery, improve resolution, and minimize those interactions which lead to sample losses. Finally, as a new products, we are going to introduce another innovative new product based on this Premier solution platform. This new system is coming soon this year, and I'm very excited. This system will also bring innovation for your success again. Today, I gave you a brief overview of Quorant Waters' LC portfolio. Thank you very much for your time and attention. Please contact your Waters representative at any time if you have any chromatography questions. Waters has a system solution to help you answer the questions with confidence. Thank you again for listening.
Thank you, Charlotte, for the great overview of our current LC portfolio, especially our latest addition to the family, Premier LC, the state-of-the-art technology that we expect many of the pharma labs will find it useful to solve some of the current consistency issue related to metal-sensitive compounds. Next, I would like to welcome our next two speakers, Dr. Pauline McGregor of PMAC Consulting, as well as our very own Dr. Paula Hong from Waters. Pauline will walk us through a process called Instrument Quality by Design, which assesses risk and employs both control strategy which, when applied, can mitigate variability and ensure data quality when switching your validated LC method to either a different system or geography. Paula will help to demonstrate this principle by examining key challenges faced in a routine analysis laboratory and showcase successful method transfer to 4 different ARC HPLCs in labs across the world, highlighting the robustness and high quality data obtained from ARC HPLC. I'll now hand it over to Pauline.
Hello everyone. Today I'm going to introduce a proactive process that can be used to help identify, control, and reduce the risks of using different LC instruments during method transfer or even if you're considering switching your validated procedures to new or an upgraded instrument. The process is based on the principles used in quality by design and lifecycle management. When considering a new or upgraded LC system or preparing for method transfer of a validated procedure between different vendors or models, the following questions are often asked. How will this new, different instrument impact the performance of my validated analytical procedure? And will it work? Well, the expected and dreaded answer is, of course, it depends. Here you can see a general diagram of the QBD lifecycle management process. This process can, in fact, be applied to any process, since a process basically boils down to inputs and outputs and understanding the variables and interactions in between. Application of this approach in the pharmaceutical industry became popular with the publication of ICH-Q8 in 2005. Followed by its companion documents ICH-Q9 and ICH-Q10, and then later ICH-Q11. It was recognized that analytical procedures could also benefit from a similar approach, hence Analytical Quality by Design, AQBD, was conceptualized around 2007. As we know, performance of the instrument is the foundation of any analytical procedure and the generation of quality data, so it is important to have confidence that any instrument will perform as required. Yet, when instruments are referred to in AQBD, it's usually in the context of a prerequisite that instruments are properly calibrated and qualified prior to proceeding with the validation of an analytical procedure. Given the problems encountered in the industry when running validated procedures on different LCs, it does not seem like equipment, calibration and qualification are enough. Let's have a look at how using a quality by design lifecycle management approach will help proactively identify and control potential problems due to differences in LC models when running a validated procedure. So what is instrument quality by design? IQBD, a systematic lifecycle approach to optimize instrument performance suitability between instruments that begins with predefined objectives and emphasizes instrument understanding and control based on sound science, knowledge, and quality risk management. It creates a succinct and proactive approach to understand the risks to validated analytical procedures when considering or faced with using a different model of LC. The process is independent of test procedures and focuses on instrument performance. It detaches instrument problems from all the other variables that exist within an analytical procedure, thus instilling confidence that any problems that may occur with the analytical procedure in the future will not be due to a change in instrument. The workflow looks something like this. The first step, as with any QBD process, is we have to define a target up front. In this case, we determine the instrument suitability target, the IST. The next three steps, again, are typical of a QBD process, Stage 1 lifecycle management. We gather knowledge, we perform a risk assessment, and then we define and apply a control strategy. And then we can move on to Stage 2 lifecycle, where we demonstrate instrument suitability. We do not cover Stage 3 and 4 of the lifecycle management, since Stage 3 is already covered in our current quality systems within our maintenance and calibration programs. And Stage 4 is, of course, retirement replacement of the instrument, which would lead us back to Stage 1. This is an example of what an IST could look like. Instrument performance is generally measured in terms of peak response, so peak shape, resolution, retention time, sensitivity. As you can see, some of these key factors are captured with acceptance criteria in our example of the IST. Gather knowledge. This fishbone illustrates the operational parameters of any HPLC that could impact its performance output. By mapping the process of the components of the HPLC system, we get our main bones or section of the fishbone diagram. For example, solvents delivery system, injector, detector, and measurement data system. I have included two additional sections, materials and system environment. Although not part of the HPLC itself, these are particularly important since they reflect end user or real-time field information, which is known to vary widely. So it's important that we consider these in our knowledge gathering. Prior to gathering our knowledge and performing a risk assessment, we need to define a risk statement. Our risk statement, in this case, is identify instrument features which, if different, could potentially impact the performance of the instrument to the extent that the requirements of the IST would not be met. We can simplify the general fishbone on the previous slide by including only the components that are different between the systems under comparison. We can remove similar instrument functions. We find out this information by comparing the specifications. And we can remove things that we can control in our environment. The example we're going to use today is a customer considering the upgrade from an Alliance 2695 to the ARC HPLC. So if we apply the customization of the fishbone according to these steps, this fishbone simplifies pretty extensively due to both systems having the same measurement system and the detector, so these can be removed. And also it has some identical design mechanism between the systems, such as the injector, the column oven, and the sample compartment. The instrument suitability evaluation will use the same column and vials, so these can also be removed because these will be controlled. Based on our simplified fishbone, we can begin to gather knowledge against our risk statement and prepare for the risk assessment. Remember, the outcome of the risk assessment is the control adjustment strategy that we will apply to accommodate any instrument differences to ensure the requirements of the IST are met. It is important to note, as with any risk assessment process, the effectiveness of the control strategy is dependent on the strength of the knowledge gathered and the accuracy of the allocation of risk levels during assessment. So on this slide, we continue our quest for knowledge, excuse me, and it's really important to know your current system. So we observe, we measure, experiment, and document. We measure the document dimensions and material of tubing connecting the injector to column, the column to the injector, and the flow cell tubing. Variation in tubing is a common culprit of differences in instrument performance between instruments, and this is because the tubing material and dimensions impact system volumes such as extra column volume and 12 volume. So we want to note and document the types of material of any connectors and fittings as well. Then we want to run experiment to determine the actual extra column volume and dwell volumes on our current system. We need to document flow cell path lengths and volumes as well, so we can compare them to the second system. Finally, prior to performing our risk assessment, we need to choose a risk tool and set predetermined risk criteria. That's just a reminder of our risk statement. Our predefined risk assessment criteria, we're going to use a heat map as our risk assessment tool, and we're going to define our high risk in red, which indicates that further knowledge or experimentation is required as it will impact the IST. Our medium risk will be yellow, which indicates that further knowledge or experimentation may be required as it may impact the IST. And our low risk will be green, which indicates that further knowledge or experimentation is not required as it will not impact the IST. So armed with our risk statement, our predefined risk assessment criteria, our customized fishbone, we're all set to perform our risk assessment. Our knowledge gathering then allows us to assess any remaining differences against our risk statement. The first column you see in this table, this is where we list all those bones, these remaining bones or the potential impact variables from our customized fishbone. Then we use our predefined risk criteria to assign risk levels to our particular scenario. It's also useful at this stage to know a bit about the analytical procedure for which the different instrument will be used. This is easy for method transfer as we know the analytical procedure that we wish to transfer. When using this tool for assessing the use of new or upgraded instruments, then a typical procedure or procedures which will be run on the new or upgraded instrument can be considered, like what will its intended use be. This process allows any unique analytical procedure requirements to be evaluated. For example, if our particular procedure requires low injection volume, potential solvent effect due to mobile phases, or in particular analytes, then this can be considered in a reiteration of the knowledge gathering and risk assessment process. Let's take a closer look at the risk table for our example. A typical intended use of the ARC was going to be for isocratic and gradient methods used for small molecules. If you look at everything in green, although these potential variables have been identified as differences between the two equipments, we've designated the green as or the low risk areas, and we've justified why they're low risk. For example, minimum sample requirement is different between the instruments, and they could impact precision and sensitivity. However, the analytical procedure that will be run on the ARC do not typically require low sample volumes. Again, the injection accuracy, injection precision, although different, then both instruments report values and criteria that are well within the intended purpose of what the new instrument will be used for. Due to the information gathered in this particular scenario, the differences in dwell volume and extra column volume were identified as a medium risk, and because the tubing had been changed on the original aligned system currently in use, this was identified as high risk, as it may impact system volumes. So from this risk assessment, we can come up with a control or adjustment strategy. Before we do that, we need to document our risk decisions, which we have in the table, and also I've added, we want to add high and medium risk to our control strategy, and we don't feel confident that the mixer will be high risk, so we want to gain some further knowledge. Our control adjustment strategy for this scenario would look something like this. We want to ensure analytical test methods have method parameters that run within the operating ranges for both instruments. We accommodate any differences in system tubing at time of instrument installation. We will adjust for differences in dwell volume and accommodate any physical adjustment required for mixing inconsistencies at the time of installation. For method transfer, the control strategy can be applied during the gap analysis prior to conducting the method transfer. For new equipment, it can be requested to be applied during the installation process as part of the user requirement specification. After we have accommodated the control or the adjustment strategy, we're then ready to move to stage 2 of the lifecycle management and perform the instrument suitability evaluation. This is run using quality control reference materials as the instrument evaluation test solution on both instruments. We then compare the output from each instrument, and it should meet the IST based on the results obtained from running the QCRM. The benefits of IQBD is that it allows us to understand and gain knowledge about your current instrument. It allows us to focus on the relevant performance technical aspects of instrument specifications. It helps us detach instrument problems from other variables that exist within an analytical procedure. It can help predict potential problems due to differences in instruments and take a proactive approach to prevent these problems. We demonstrate instrument suitability through a documented process against predetermined acceptance criteria. And this will provide confidence that any problems that may occur with the analytical procedure in the future will not be due to a change in instrument, which allows us to enjoy advantages of successful method transfer or updated technology without having to revalidate analytical procedures due to instrument performance differences. In summary, IQBD can be applied to different models or vendors of LC systems to assess suitability. It can be applied when assessing the switch from HPLC to UPLC systems. And it can be used in method transfer as part of a gap analysis prior to the transfer of analytical procedures to help remove any potential problems due to instrument differences. The complexity of the IQBD process will depend on the types of differences between systems and the necessary adjustments or controls required to accommodate them. That's all I have for you today. Thank you very much for your time and attention.
Hello. Today we are going to discuss the instrument suitability evaluation of HPLC systems looking at a USP organic impurities method. This study will entail moving a method from one instrument to another type of the same instrument in a single lab, as well as moving that method to many other different labs around the world. What are some of the considerations for adopting modern LC technology in a global economy? Well, new instrumentation is often desired to deliver sustainable competitive advantage. New instrumentation can have improvements in both performance and usability. However, adopting these instruments into our lab can result in a diversity of instrumentation in a single site. Furthermore, there is often a challenge to increase profitability and efficiency. For this, we need to harmonize our approach across many sites and simplify transfer from one lab to another. However, with that comes the requirement that we need to manage a diversity of available platforms. To perform this work, we outlined a methodology that we wanted to use. The first step included identifying an appropriate validated method. The preference was for a method that we had previous experience with in our laboratory, as well as ensuring that the method variables would not impact the system-to-system or site-to-site reproducibility. Once the method was identified, we then looked to analyze the method on two types of different systems, or HPLC systems, in a single site. Prior to performing that experimentation, we want to make sure we correctly assess the impact of the instrumentation and develop the appropriate control strategies. Our last step was to transfer the method across many different sites globally. Prior to doing this, we also performed another risk assessment and developed a control strategy. We then identified the laboratories, sent the required materials and consumables to each laboratory, and had a strict procedure on collecting data and performing data analysis. We identified the USP monograph for quetiapine's fumarate impurities as the method we would choose. The systems were identified were the Alliance with an IEEE cable connection to the computer and a 2489 TUV detector, as well as an ARC HPLC with a 2489 detector. Some of the key characteristics of the method include a high pH mobile phase at a pH of 9.2. It is a gradient method with an atypical gradient and delivery of 70.7% B, as well as a system suitability mix readily available for purchase and a drug substance that we had purchased many years ago. While it was past the expiration date, we had enough of the drug substance to readily ship around the world. The column of choice was the Expert C8 column, 4.6 by 150. Other critical method conditions included the column temperature, which was elevated above ambient at 45 degrees Celsius, and a flow rate of 1.5 mL per minute. As described earlier, we purchased the samples in standards, although the drug substance had been purchased quite a while ago. And we also used the system suitability criteria as assessment of our analysis, including the system suitability criteria for the system suitability solution, which included resolutions of 2 critical pairs, as well as that of the standard solution, which included tailing, retention time percent RSDs, and area percent RSDs. So for the first part of our study, where we looked at running the same method on 2 different instruments in the same laboratory, we developed a control strategy. As we outlined earlier, we determined that a TUV or tunable wavelength detector would be the detector of choice for this analysis. We wanted to also ensure that we use the same flow cell path length as well as type. Because the sample managers operate slightly differently, we wanted to ensure that the wash solvent on the Alliance would be equal to that of the strong needle wash on the ARC HPLC, to reduce any differences we may observe in sensitivity due to carryover. Lastly, we wanted to ensure that both of our instruments provided adequate temperature control. As you recall, the temperature for the method was 45 degrees Celsius. So for that reason, we ensured that the passive preheater was installed in both systems. And lastly, because we were looking at instrument-to-instrument variability, we had a single analyst perform both analyses. Here are the system suitability results for the analysis performed on both the ARC HPLC and the Alliance IEEE. Shown on the left is a system suitability solution. For both the system suitability solution and the standard solution, both systems met all of the USP specifications and were well within the requirements. This included resolution of quetiapine and quetiapine disoxy, resolution 1, as well as the resolution between related compound G and related compound B, which are peaks 1 and 2. Furthermore, the tailing factor, retention time RSDs, and area percent RSDs were well below the limit of not more than 5%. Looking at the drug substance results, which were really the intent of the analysis, we observed comparable quantitative results on both the Alliance IEEE and the ARC HPLC system. Both the quetiapine disoxy and an unknown impurity were observed in both analyses looking at the drug substance. And measurement of both gave very similar quantitative results in terms of not more than meeting the acceptance criteria of not more than 0.15% for the quetiapine disoxy and not more than 0.10% for the unknown impurity. Furthermore, both of the total impurities were well below the limits. It should also be noted that this drug substance was well past the expiration date, and therefore we would not have been surprised if the impurities levels were significantly higher. After completing our intralab study, the next step of our analysis was to perform an interlaboratory study, looking at the method performance across many ARC HPLC systems. Again, our objective was to replicate the analysis for drug substance across many different laboratories. However, we used a single type of instrument instead of multiple types of instruments. We again use the same method as described earlier. However, this time our ARC HPLC system could have either a TUV 2489 detector or a photodiode array 2998 detector. This was because of the availability of the detectors in the different laboratories varied. Again, we were going to assess our results by ensuring that we can meet our system suitability requirements as described in the monograph, as well as analyzing the sample formulation or drug substance and comparing the impurities analysis obtained. As we described earlier, many of us in the laboratory had experience with this method. So with that in mind, we were able to perform a risk assessment internally. We focused on some of the key attributes that might impact method performance, including sample prep, the materials, the method, and the instrumentation. Outlining these factors, and with the typing fumarate organics impurity method in mind, we identified some key characteristics or components that could impact the long-term method performance of the analysis. This included the pH adjustment, since the mobile phase pH was required to be at a pH of not less than 9.2. Furthermore, impacting the mobile phase pH could be the quality of our materials, including the ammonium acetate and the ammonium hydroxide. Other method conditions that we identified as critical for method performance, including the column temperature or temperature control, because of the elevated temperature conditions of the method, as well as the gradient table because of the unusual numbers required to enter into the table. Lastly, we identified the detector. As we described, we allowed labs to either have a PDA or TUV detector, but that also added a variable that we needed to risk assess. With the risk assessment performed, we gathered as a group to try to rank or score the different failure modes that could impact our method performance. We first identified the failure mode, the source, as well as the type or failure effect. In our discussions, we realized, going back to previous studies, that an analyst had performed robustness studies looking specifically at the acetonitrile buffer ratio in our mobile phase and how it could impact our retention time and system suitability. In her studies, they were found to have a dramatic impact. So with that information and looking over the critical failure modes, we identified two that were more likely to have an impact. These were the acetonitrile buffer ratio and errors that could be occurring because of the use of certain graduated cylinders or in preparation of the mobile phase, as well as the filter vacuum equipment that was available. Excessive vacuum could have impacted the acetonitrile buffer range over a long-term period of time due to the evaporation of the organic portion of the mobile phase. So those were two of the critical areas that we determined needed to be controlled. So with that in mind, we wanted to ensure that there were control strategies set up to ensure the reproducibility of the method around the world. With that in mind, we looked at logistics. We shipped a single chemical kit from our Milford, Massachusetts office, including standards, columns, and formulation. Note, not all of the columns were of the same lot. We then compiled a standard operating procedure protocol that would be distributed to the many labs, baseline the method in our lab, and then transferred the method to another laboratory within our site, as well as Turkey, India, and Singapore. The identified areas of concern are that we wanted to make sure there would be no changes to the USP method. So with that in mind, we described careful preparation of the mobile phase, including filtration of the mobile phase, and the use of new or recently opened containers of ammonium hydroxide and ammonium acetate. So this is what our protocol or standard operating procedure looked like. We wanted to ensure that the descriptions and the instructions were clear and easy to follow. So with that in mind, we had our Milford, Massachusetts laboratory acting as our beta site as well for the SOP, providing commentary and asking any additional comments in areas where there may have been clarification needed. So once we got the comments back and revised the SOP, we sent it to the other receiving laboratories. In addition to the SOP, we wanted to get information on the instrumentation and configuration that all of the laboratories used around the world. So here is an example of the information we asked for, including the pump, auto sampler, column heater types. As described, we specified that a passive preheater was required, but again, we wanted verification of our instructions. The detector could have been TUV or PDA, so that information was critical, as well as verification of the flow cell. And lastly, we didn't specify what type of tubing needed to be placed from the column outlet to the detector, so that was one key piece of information we asked as well. When we got back the information, we did see one unexpected configuration in the lower ID, 4000s ID peak tubing, which was not standard for this system. However, we just kept that in mind when looking at the results. So these are the results we obtained chromatographically for the system suitability solution from the many labs around the world. So what is very impressive is we got very good retention time reproducibility. It is more impressive given the fact that we know mobile phase preparation can have a significant impact on the reproducibility of the method. So obviously the SOP provided adequate instructions for the new chemist or analysis not familiar with this assay to be able to perform it reproducibly. All of the sites met the USP resolution criteria for the system suitability sample for both resolution 1, which was between the quetiapine and disoxy, peaks 3 and 4, as well as resolution 2, which is in between peaks 1 and 2. With the information we had about the different ID peak tubing used between the column outlet and the detector, we also went back and looked at the system pressure traces, which were collected by all the laboratories. As you can see here, the laboratory that used the lowest ID tubing, or 4000s ID, had a significantly higher back pressure than the other laboratories. In addition, we saw the narrower peaks observed when we compared the chromatograms. However, the resolution criteria were very comparable, as well as additional results, as you'll see later on in this presentation. To evaluate the data beyond meeting system suitability specifications, we want to ensure we were in a state of control. So we developed a control chart to look at repetitive processes and monitor process variables. Our requirements for our state of control was plus or minus 3 standard deviations. Here are the control charts we used for the system suitability solution, specifically looking at resolution 1 and resolution 2. As described earlier, all of the results from each different laboratory easily met the USP system suitability specifications of resolutions not less than 4 for resolution 1 and not less than 3 for resolution 2. However, furthermore, they were in a state of control, all within 3 standard deviations of the results observed. Part of system suitability also requires the analysis of the standard solution for tailing factor, retention time percent RSDs, and area percent RSDs. So as you can see on the left-hand slide, all of the chromatogram retention times line up very well. The standard solution is at 1 microgram per mil of quetiapine, and all of the system suitability requirements were well within the specification, including the tailing factor of not more than 2.0, the retention time RSDs of not more than 5%, and the area RSDs of not more than 5%. So if we look at the control charts again, we can see that all of them are well within control. It is important to note that retention time RSDs are so much lower than the specifications that the control limits are very hard to see. However, the retention time reproducibility was very consistent. When we looked at the area RSDs, though, we did notice some outliers. 2 of the points seemed to be slightly higher than the other 3 points in our study. So we wanted to look a little bit more deeper into that information. We wanted to do a deeper dive to make sure we understood the variables that could be causing the higher percent RSDs. So with the information we had obtained from the labs, we realized that the two laboratories that had higher percent RSDs both used a PDA detector. Now the percent area RSDs were well within the specifications. However, the areas were well above 1%, which was higher than we observed with TUV. Looking at the chromatograms, we can see that there are more perturbations in the PDA data than the TUV data. This is not surprising, as a TUV has higher sensitivity and a wider linear range than photodiode array detectors. However, many laboratories choose to use a PDA because they can use peak purity and library matching and get spectral information of their peaks. With that in mind, we can see that the TUV did perform better for this analysis. However, both detectors met the specifications. This is a useful strategy if you are looking to assess any variability in your cross-laboratory results and understanding why they may be observed. Realizing that all of the labs met their system suitability criteria, we then looked at the sample solution or drug substance and the quantitative results. Again, the retention times were all matched up very well, with relative retention times of within 0.02. Furthermore, all of the labs got very comparable percent quetiapine disoxy and unknown impurity. As you can see here, they met the acceptance criteria of not more than 0.15 for the quetiapine disoxy and not more than 0.10 for the unknown impurity. Again, because the drug substance was very far past the expiration date, we were not surprised to see impurities present in this formulation. Again, looking at our control charts for our sample solution, we got results that were well within control, very far below the 3 standard deviation and below the specifications required for analysis. In summary, we have shown that the USP impurities method for quetiapine fumarate can be successfully run on a variety of instruments, both within a single lab and across the globe. Understanding of our system and our configuration provided greater control and increased the likelihood of meeting our system suitability criteria. Furthermore, understanding risk of our method and implementing control strategies led to improved reproducibility and system performance. Lastly, through this interlaboratory study, we were able to demonstrate the ability to successfully replicate USP monographs globally on the ARC HPLC system. This work could not have been possible without a very large group of individuals helping and supporting the efforts. So here are some of the many individuals that helped support the process and experimentation, both in our Waters, Milford, Massachusetts site, as well as around the world. Thank you for your attention.
Thank you, Pauline and Paula, for those informative talks. Our next two topics will be presented by Jennifer, the Director of Product Marketing for Chemistry Technology Center, and Xiaodong, Biopharma Marketing Manager from Waters Corporation. Jennifer will demonstrate how to increase repeatability of mundane tasks prone to human error by harnessing the power of automation and quality consumable to increase accuracy and avoid costly out-of-spec results. Following that, Xiao would address the challenge of non-specific adsorptions with our newest premier technology, giving specific example of 3 classes of charged species such as peptides, glycans, and oligonucleotides. Here is Jennifer to take us through her first presentations.
Hello everyone, and welcome to the talk today around reducing variability with quality tools and simple automation. My name is Jennifer Fournier, and I am the Director of the Chemistry Product Marketing Group, and I want to thank my colleagues Jonathan Turner and Paula Orens for their contributions to this presentation. Here are some statistics to ponder. Over 80% of laboratories operate in a regulated SOP, standard operating procedure, driven environment. And it's estimated that over 35% of irreproducibility has been attributed to manual errors in performance of experiments and data reporting. And this has cost an estimated $28 billion in terms of having to repeat experimental work. The fact is, we work in a regulated environment that requires reproducibility, and that leads to many laboratory challenges. Our assays have to be repeatable and accurate. Our protocols need to be reliable, and they need to be reliable whether we transfer them to another user in a lab or we transfer them to another site globally. We require traceability and compliance, and we strive for productivity and efficiency. In late-stage development, the expectation is that the separation methods created will be registered, controlled, and will be supported throughout the lifetime of the product. Understanding key parameters that will impact the goals of your method early on should help avoid costly method reworking and rerunning of samples. And that is what we're going to discuss today. Who likes unexpected results? Not me. So we want to avoid unexpected results at all costs. Often when an unexpected result happens, investigations into the materials used and/or the analytical method will take place. Is it the manufacturer or supplier's fault? If there is uncontrolled variation in the vendor who supplies the testing material, then it is impossible to have confidence in results. Likewise, if there are questions around the method's robustness, it's also impossible to have confidence. It's important early in the process to partner with a vendor committed to quality. It's also important that your method development process follows a quality by design approach, so you understand the key parameters that impact the method. We are going to take a journey today, and the goal of that journey is to get the sample to the result. Each of the steps of the sample journey introduce risk and potential variation, which needs to be looked at. These risks can come from the workflow itself or the consumable products used. The analytical method is a vehicle, so to say. It is the method that carries the sample through the workflow and ultimately ends at the result. So the first part of our journey that we want to prepare for are factors that impact reproducible sample preparation. Many of you who have worked in a laboratory have most likely done either or both of these common manual laboratory tasks. They probably look very familiar. The tedious documentation in a laboratory notebook is on your left, and repetitive pipetting is on your right. Both tasks are relatively simple, but critically important to be accurate. Assay repeatability, assay accuracy is at the core center of both of those tasks. Laboratory managers must ensure protocols and workflows are carried out according to documented procedures. And standardizing the analyst technique can help improve compliance with assay specifications, and these minimize errors. New technology, including connectivity and automation, can help improve assay repeatability. Let's take a look at standardized protocols. The fact is, the majority of results generated with manual manipulation are still captured via paper and susceptible to error. Standardizing digital protocols with connected laboratory devices simplifies the process of harmonizing laboratory procedures across users, labs, and sites. Protocol standardization improves data comparability while removing the ambiguity of pre-analytical techniques. User and lab management ensures that only approved procedures are utilized. This minimizes nonconformities. Leveraging software, connected tools, and guided protocols eases workforce training and improves laboratory outcomes during error-prone tasks. What if you could even show a video within your protocol? You could capture many different ways an analyst learns. Now let's talk about traceability and compliance. Improving traceability and compliance really can assist in the avoidance of costly errors. With protocol automation, you can enable consistency, accuracy, and traceability of workflows, especially with complex error-prone tasks like pipetting and sample preparation. Additionally, automatic electronic documentation of activities improves the availability and legibility of experimental records, making it easy to see exactly what happened at each time point within an experiment. This is especially useful for evaluating out-of-spec results. Smart connected tools can help do all of that with timestamps, analyst IDs, and equipment calibration records. Here are some tools that we have at Waters that can help aid in all of this. The Andrew Alliance Connected Lab ecosystem allows you to do automated pipetting driven by powered by electric laboratory methods that are easy and intuitive to use. Andrew Alliance offers connected laboratory products that enable seamless execution of sample preparation protocols. Andrew Plus is your pipetting robot. It can automate workflows, and you can do more complex workflows with its connected device ecosystem. What does this do? It improves efficiency, reproducibility, and it offers, through the OneLab connected software, the benefits of experimental traceability. We also offer Pipette Plus. This is the electronic pipetting system that Andrew uses that can be on the Andrew or as its own laboratory system. This helps to ease the burden of manual sample preparation by improving productivity and reproducibility and leveraging the OneLab protocols. OneLab is at the heart of all of this. That is your connected laboratory software that we can design protocols, share them across users and labs, and execute them across a variety of connected devices. Its user interface is designed to make it easier than ever to execute sample preparation workflows. Let's take a closer look at the OneLab software. This is a browser-based software that can run on any tablet or PC. You could do it right now. You could simply log on to onelab.andrewalliance.com and start to create a protocol that is really common in your laboratory. OneLab enables the design of sample preparation workflows through an intuitive user interface featuring a drag and drop functionality. It's super easy to pick up and use. It's easy to perform complex procedures as well, including serial dilution and concentration normalization with the wizards that are embedded in the software. Protocols designed in OneLab can be executed both in manual mode as well as with any connected device associated with your OneLab account. The other tool that we have in our toolbox is the Auto Specialist. The Auto Specialist is our new positive pressure manifold, and this was really built to minimize user-to-user variability and improve reproducibility when it comes to SPE. The new Auto Specialist processes a wide variety of different formats, such as 96-well microelution plates and 1cc, 3cc, and 6cc cartridges. It too is controlled by intuitive software, the Auto software, which is CFR Part 11 compliant, and it's also easy to use. It allows you to program pressure control for each step of the workflow, as well as assay methods, which combine the pressure profiles, manifold commands, and user alerts. These methods can be used by various scientists and shared between lab locations easily. A software-enabled positive pressure manifold really changes the game in terms of SPE reproducibility. Now let's go on to our next part of the journey, which is consumables and method development approaches. Well, we now have our sample and it's been logged in to our software. So the next thing that we need to do on this journey is have a robust chromatography method. So what does a robust method mean? It may be obvious to some, but I'm sure that there are different answers to this simple question. Ultimately, it's having confidence in that result generated. And that can come from robustness in regards to changing solvents, consumables. We don't want that to change the confidence in the results. We don't want the method to be open to interpretation. We want to be able to easily pinpoint the cause of method failures during investigations. And most critically, we want the least amount of out-of-spec results and out-of-trend results. Three major factors help instill that confidence in the results. These are consistent sample handling, which we just talked about, consistent consumable performance, as well as consistent instrument performance that we're going to talk about next. So what's important when selecting consumables? Consumable products are a key part of your analytical workflow, and it's important that the consumables that you select meet three specific application criteria. They need to be fit for purpose. You want the right product for your method. They need to be reproducible. We need batch to batch, device to device, and column to column reproducibility. And we need to rely on our vendor, quality and dependability. Especially nowadays, supply chain is so important. Arguably, the most important consumable in a chromatography method is the column. So let's take a look at analytical LC columns and what to look for from an LC column vendor. If we look at column manufacturing as an example, there are three key criteria that enable a robust, reproducible column. There's the hardware, the materials and the design, the sorbents, the chemistry and the synthesis process, as well as the optimization of the packing. And Waters looks to ensure that all three of those things are at the highest quality. The end game is confidence in your results. In partnering with the right consumables vendor, one that's dedicated to controlling quality and implementing a quality design method development approach, you can have reproducible separations over the life cycle of your method. Here's an application note that shows a separation of an analyte using 10 columns, each packed with material that spans over 15 years of manufacturing. Now that's a long time. And as you can see, the retention time and relative retention time variability is very low. This provides confidence in the result over a long period of time. The next part of our journey that we want to discuss is the best consumable for your system. What column configuration is best for your system needs to be considered because the fact is not all systems are created equally. There are many LC systems on the market, and most of these LCs are column agnostic when it comes to the analytical columns that you can attach to them. For older HPLC, the typical column is 4.6 millimeter in internal diameter, packed with greater than 3 micron particles. However, in recent years, as the column technology has improved, improvements to the system have also been made to realize the performance benefits of high-performance LC columns. Now, it's very critical to understand the LC system, specifically what impact it contributes to the separation. So let's take a closer look at what makes the LC systems different. Instrument dispersion is really at the key of it. Every LC system contributes to the broadening of the analyte band. It's an individual system dispersion value that can be measured to identify exactly how much volume the system is contributing. It's important to classify the LC system by this value. Typically, HPLC systems have high dispersion values, where UHPLC have very low dispersion values. Selecting analytical columns that best match these values is critical to achieving maximum chromatographic performance. Let's look at the example. System dispersion, the combined effects of tubing and its connections, sample valves, flow cells, and column end fittings, causes the sample peaks to broaden owing to dilution. That begins at the injector and ends at the detector's outflow. As you can see, as the size of the particles in an LC column are reduced or the internal diameter and length of the column is decreased, the potential peak broadening in non-optimized LC systems increases. Here we have 3 columns with different internal diameters packed with 2.7 micron particles. We've run the sample separation on each column using an Alliance HPLC, which has a high dispersion value. As you can see from the chromatography, as the column ID is reduced, the peak shape and resolution get worse. Some may think it's a column, but it's not. The band spreading from the system is swamping out the efficiency. Larger columns, typically 4.6 millimeter, perform best on this system because the ration of the column volume to system volume is more favorable. The higher column volume is less impacted by the fixed system volume. Now let's look at matching the system with the proper column internal diameter. If we take those same 3 columns and run them now on 3 different systems, in HPLC, UHPLC, and UPLC, you will see the columns were not broken, and you now get the expected equivalent performance on the 3 systems. We now must match our column internal diameter to our system, all based on dispersion value. We have categorized LC systems into 3 categories: HPLC, UHPLC, and UPLC. HPLC has a dispersion of 30 microliters or greater, UHPLC sits in the middle, and UPLC has a dispersion level less than 12 microliters. Due to these dispersion levels, we must appropriately match the right type of column size, volume, and particle size with the system dispersion to maximize column performance. A column with 3.5 and 5 micron particles can be used on HPLC systems, while the 2.x micron particle columns are appropriate for UHPLC systems. And columns with sub-2 should be used on a UPLC system to gain the best efficiency and best resolving power. The last step of this journey is ensuring that the journey is smooth for the long haul. We need to make sure that we assess LC system health, and we can do that with the help of standards. So how do we use a reference standard to assess system health? Well, a reference standard is any standard that is used independently of an assay method to assess system health. It can be made by the analyst or purchased from an outside member. The reason the reference standard is not the system suitability standard is that if a system suitability standard fails, this often leads to a report and investigation as to why it failed. By using a reference standard to assess system health, a failed RS may not trigger an investigation or any additional paperwork. Additionally, for those customers not in a highly regulated environment, an RS may be their only standard that they run. This is especially true in method development laboratories where running standards is not required. So the use of a reference standard can help those analysts gain confidence in their instrument before performing method development or any other experiments. There are 3 ways to use reference standards. You can use a reference standard to benchmark. This is when you want to know that the system is in a good state, maybe after installation or performance maintenance. For qualification, you can use a standard to do routine qualification checks to confirm continued good system performance, or maybe use it when a failed qualification check would trigger troubleshooting. And finally, for troubleshooting, using a standard to troubleshoot system problems and check if repairs are made and have fixed the problem allows you to have the same conversation by using the same reference material with whomever you're troubleshooting with. Here's an example of how a reference standard would work in a typical workflow. The example here is incorrect mobile phase preparation, and we're using our reverse phase QCRM. Before we started running system suitability and our precious samples, we'd first run this quick system check to ensure that the system is in good working order. We're going to compare the data against a known benchmark, which is the reverse phase QCRM, and as you can see in this example, the check would quickly indicate that the system was not ready for routine use, and some troubleshooting would need to be performed. After a remake of our mobile phase, you can see that we are now passing our specs and we can go on to running our system suitability and our precious samples. To wrap up, remember the journey. In order to have confidence in your results, which is the end game, automation can deliver improved efficiency, speed, and reproducibility and lower the cost by replacing manual methods. Using automated pipetting helps to increase accuracy, consistency, and efficiency, and the more details that are put into the method, the better. And with connected devices and software, such as you saw with OneLab and the Auto Specialist, everything can be documented. You want to choose consumables and consumable vendors that can ensure product quality and reproducibility. This can help avoid costly out-of-spec results. Be aware of the contributions your LC can add to your chromatography. Matching the LC column internal diameter with the class of LC systems is really important. And finally, using reference materials to monitor your LC health will help to ensure that your LC is in good working order prior to injecting your precious samples. So thank you for joining me on this journey, and hopefully you were able to take some learnings from this to make sure that in the future you have confidence in your results.
Hi everyone, thank you for attending this session. My name is Xiao. I'm from the Waters Biopharmaceutical Business Team. Today I'll go over some of the Waters ACQUITY Premier Solutions application in biopharmaceutical LC analysis and show how this solution could really bring benefits to your laboratory operations. I'm very excited about this new technology and solution, so let's get started. Having looked at the general introduction of Qt Premier technology, I'll go over why it is the ideal solution for biopharmaceutical analysis. Because the Qt Premier solution can minimize non-specific interactions and absorption, the direct outcomes are improved analyte recovery and better peak shape and resolution. In addition, because this solution can achieve optimal performance without passivation, cleaning, or conditioning, it also reduces method-related variabilities due to different ways these procedures are performed in different settings. Overall, the solution really brings benefit to the analytical operations by enhancing data quality and operational efficiency. In the following section, I'll show how Premier technology can improve the recovery, resolution, and MS data quality of three classes of analytes: protein and peptides, glycans, and oligonucleotides. It is well documented that the PanK peptide is susceptible to post-translational modification such as deamidation and contains 4 negative charged amino acids. Furthermore, the PanK sequence is in the constant region of the Fc domain in the humanized antibodies and is often a product quality attribute that must be monitored. On a conventional LC column, only one of the product-related impurities was partially resolved from the main peak due to excessive tailing. The tailing factor is calculated to be 2.74. On a Qt Premier column, the tailing of the main peak is reduced due to minimized non-specific interactions. As a result, improved separation reveals the presence of 2 product-related impurity species. Both are well resolved with baseline separation. The tailing factor here is calculated to be 1.25. This example demonstrates the value of max-peak high-performance server technology can bring to the lab by improving the analysis of trace-level product-related impurity peaks in monoclonal antibody therapeutics. Now let's look at how Qt Premier system and column can work together to improve reverse phase LC separation recovery and MS data quality. The example here shows the reverse phase LC-MS height and area response data of an acidic peptide. On conventional metal-based LC system and column shown in orange colors, we're seeing limited MS height and area response. This is due to non-specific absorption and interaction of the analytes with metal surfaces. The results from a second experiment, represented by green colors, are showing improvements when conventional column is replaced by an Qt Premier column, resulting in 16 times better MS height response and 2 times better area response. In the third experiment, showing blue, even more dramatic improvements were achieved when both an Qt Premier column and a Qt Premier UPLC system were used. We achieved 27 times better MS height response and 4 times better MS area response. The results here show that the overall Qt Premier solution, combining high-performance surface-based column and LC system, can work together to maximize the performance of separation and MS data quality. Increased recovery is important for MS sensitivity, but also increase the confidence in data quality. In this experiment, data-independent acquisition created 11 BY peptide fragments associated with the T14 peptide of a tryptic digest of the NIST monoclonal antibody reference material. The MS data was obtained after reverse phase separation using conventional LC and a stainless steel column, shown in orange. Here we see about 90% sequence coverage. Using the same method parameters, 36 BY peptide fragments of the T14 peptide were identified for the same sample after separation using a HPS technology based on the Qt Premier column and UPLC system. Sequence coverage increased to 95% in this case. Having a look at mostly reverse phase separation of peptides, we'll turn our attention to size exclusion photography. One of the challenging aspects of MAP SEC separation is achieving good resolution and reproducibility between the main peak and the half antibody peak. In many cases, this peak may be co-eluting or partially resolved due to tailing from the main peak. Part of the contributing factor from the main peak tailing is non-specific absorption. This is where Waters Premier technology can help. This experiment looks at benefit of just the QD Premier UPLC system versus an H-class plus bio-binary system without the benefit of Premier columns. Traditionally, to address non-specific interaction problems in SCC, salt is added to suppress the interactions. Here in this experiment, the half antibody peak is better resolved from the main peak due to reduced tailing from the main peak, resulting in improvements in peak to valley ratio. This experiment shows that even after adding salt to suppress non-specific interaction on a biocompatible system, a user could further improve separation with an Acuity Premier UPLC system. Having looked at mostly protein and peptide separations, here's a closer look at glycans. The silation of therapeutic protein in glycans has a lot of physiological consequences, which has already been reported pretty extensively in literature. As a result, it is one of the most commonly monitored product, PQA or CQA. In the discussion of non-specific absorption of acidic analytes on traditional chromatography systems, N-glycan is a relevant topic because N-glycans can become more acidic due to the addition of terminal sialic acid residues, resulting in poor recovery and peak resolution during chromatography analysis. Here's an example of Waters QD Premier column can improve separation of sialated glycans. On the left figure, the sample is a sialated glycan standard separated on a conventional glycan BH amide column. Both the initial injection and injection after conditioning have shown very poor recoveries. It only achieved better recovery after additional conditioning rounds. On the figure to the right, the same sample was separated on a QD Premier column instead. The improvement here is very clear that even without any conditioning rounds, the separation has achieved pretty optimal recovery. Together, this shows the out-of-box performance of QD Premier columns. It is expected to perform right from the beginning consistently regardless of cleaning and conditioning status. The third type of molecule that we'll look at today are oligonucleotides. Oligonucleotides are synthesized and impurity created during that process need to be characterized. Although a variety of separation techniques are used, such as capillary-based methods in anion exchange chromatography, reverse phase separation coupled with mass detection has become a method of choice. Selecting the best iron parent reagent to balance resolution, sensitivity, mass detection, and ease of use can be a challenge. It is also a challenge to passivate LC system and condition the column prior to starting the analysis. Waters QD Premier is designed to solve exactly this problem. Here we present the same idea as the previous slide, showing the performance of the QD Premier oligonucleotide column right out of the box without the conditioning runs required on conventional columns. Waters oligonucleotide standard is injected onto a UPLC system. Poor recoveries of analytes were seen on the conventional column due to non-specific absorption in the first 3 injections. It achieved improved recovery in the 4th injection. On the QD Premier oligonucleotide column, optimal recovery of analytes was achieved in the 1st injection without any conditioning runs, and the same level of recovery was maintained consistently throughout the subsequent injections. Now moving to a real-world example. We look at an antisense oligonucleotide, which is increasingly becoming more common modality in the biopharmaceutical R&D pipeline. Here we show the MS signal after the separation of GEM91 on a conventional metal-based oligonucleotide column. The column had 11 injections of oligonucleotide standard before the actual sample injections. We know GEM91 elutes around 1.8 minutes, but even after extensive conditioning, the MS data shows very little signal in that region, indicating non-specific absorption of analytes by the column. Continuing from analysis of GEM91 example from the previous slide, here an QD Premier oligonucleotide column is used instead of the conventional stainless steel column used previously. The difference is a dramatic one. Without any conditioning rounds, the analysis achieved optimal recovery of GEM91 from the 1st injection and the same consistent level of recovery was maintained throughout the remaining injections. This is another example that highlights the out-of-the-box performance of Premier technology. In summary, in this short presentation, we look at the applications of Acuity Premier solution for the biopharmaceutical analysis of three classes of analytes: protein and peptides, glycans, and oligonucleotides. We demonstrate that Acuity Premier solution can improve the recovery, resolution, and the overall data quality while reducing method-related variabilities. It is a universal solution that can improve the overall efficiency of your analytical operations. With that, I hope you find this presentation informative and would like to thank you for attending this presentation.
Thank you, Jennifer and Xiao, for those insights. I would now like to welcome our final speaker, Haining from Waters Corporation, who is our APAC Regional Empower Specialist. Prior to transferring analytical method for routine use, it has to be validated. Method validation is an important regulatory requirement that can be demanding and human error prone. Haining will share with us on how Empower Method Validation Manager can help streamline your method validation processes and benefit users with improved compliance, efficiency, and reduced error. Welcome, Haining.
Hello, good afternoon, everyone. I'm Haining, and thank you for joining me today as we discuss about streamlining method validation with Empower. Before I begin, I have a question for you. How would you get a taxi today? Do you take out your mobile phone and use a mobile-based application? If you're in Japan, you're probably using Japan Taxi. In Korea, Kakao Taxi. In Southeast Asia, Grab. And in Australia and New Zealand, Uber. Being in Singapore, I naturally use Grab when I need a car ride. Ride-hailing apps have changed the way we hire a ride by offering an all-in-one platform where we can book, ride, and pay. In the same vein, how do you do method validation today? Is it time to use a workflow application that can streamline your workflow and have the potential to change the way you do method validation? Like Grab, Empower Method Validation Manager, MVM, offers an all-in-one platform for method validation where you can set up validation protocol electronically, manage all data, and process all results and validation calculations, allowing you to automate and streamline your method validation workflow. By streamlining with MVM, there are three major benefits. First, your laboratory can reach peak efficiency. Designed with the goal of optimizing traditional method validation processes, MVM is an all-in-one workflow solution that allows the entire chromatographic method validation process to be efficiently performed within Empower. MVM also provides a summary view where you can quickly determine next actions. Secondly, MVM can reduce errors associated with data transfer, spreadsheet calculations, and data integrity concerns by performing all calculations in Empower. Finally, MVM, being powered by Empower, contains the same regulatory, data traceability, and compliant-ready benefits. In addition, MVM allows validation protocol to be approved or locked in accordance to SOP. Let's dive into each of these benefits in greater detail. Imagine one morning when you were running late for work. You want to get a taxi, but your phone is out of battery, so you hail a ride the old-fashioned way. You can stick out your arm and flag or join the long taxi queue. You keep your eyes peeled on the street, waiting for a taxi to show up. And when you're finally on the taxi, you're either hoping that the taxi meter jumps lower or counting to make sure you have enough cash. If I had my phone, I will simply open my Grab app to book a ride, check for my ride's location, and pay cashlessly. Such convenience. And very much like Grab, MVM offers an all-in-one platform for method validation. If you are still going through method validation protocol paperwork, checking through validation requirements manually, calculating your validation results with third-party software packages, or struggling with the traceability of your results, MVM is the solution for you. Let's look at how MVM can drive peak efficiency in your laboratory. Rather than having your method validation SOP in a Word document, in MVM, a validation protocol is created to incorporate your method validation requirements. Here, users can set up the validation tests from linearity to robustness. Next, data acquisition requirements are entered, allowing users to specify what laboratory data is needed to fulfill validation requirements. For example, for linearity tests, we can specify that there will be 7 levels of sample concentration. Following that, acceptance criteria allows users to identify the validation target. Continuing with the previous example, the validation target can be the slope of the calibration curve, with regression coefficient of the slope having a lower limit of 0.999. With the validation protocol set up, MVM will be able to automatically track and manage your method validation. This creates a streamlined, efficient workflow, eliminating the need to rely on written protocols. With the validation protocols configured, MVM allows the user to minimize the number of chromatographic injections required and yet still maintaining result quality and integrity. Sample sets can be acquired as validation test composites using one injection for multiple validation tests. We can see that the same 3 injections can be used for repeatability, accuracy, specificity, and intermediate precision validation tests. These validation test composites help to save a total of 15 injections. The efficient sample acquisition reduces sample consumed, sample preparation time, instrument usage time, and volume of waste disposal. Even though it may be possible to acquire efficiently with the use of manual documentation or third-party statistical software, but the complexity of needing to transfer subsets of data hinders the implementation of such efficient procedures. When it comes to data reporting, the MVM default project comes with 32 report methods for quick reporting, but can also be further customized to your SOP if needed. Next, the MVM user interface offers a quick status at a glance summary. MVM guides users through the method validation workflow by clearly displaying the status of each validation test in a validation study. Simply look at the test status field and you will know at one glance what you need to do next. Besides the test status, there is automated data checking at every step of the workflow. MVM continually monitors the data for each validation test and tells you whether your data meets your validation requirements. A green check mark indicates that the requirements are met, and for data that do not meet requirements, MVM alerts the user with a red cross, while out of specification results are highlighted with a yellow hazard sign. All in all, MVM, being a total workflow-based method validation solution, along with the convenient status summary interface, allows you to perform method validation more efficiently than before. Next, we'll look into how MVM can reduce risk. with ride-hailing apps, you can hire not just taxis, but also car, motorbikes, 7-seater vans, or even buses. By doing all this in a single app, you can benefit from the convenience and not needing to consolidate data across applications. Likewise, in MVM, you can perform all the calculations needed in your method validation workflow, saving time and reducing risk associated with data transfer. Let's look at how this works. Method validation often requires users to perform both chromatographic result calculations as well as the statistical calculation of validation results. Currently, most companies perform the latter by exporting chromatographic results to a third-party software package. However, data transfer from CDS to spreadsheets require manual verification to eliminate transcription errors. With the diverse validation calculations required, it can be daunting to develop and validate many formulas on spreadsheets. Calculations using spreadsheets often pose data integrity and security concerns. So what if you can do both sets of calculations all in a single application? MVM can eliminate the concerns of transcription errors, inaccurate spreadsheet formulas, and inefficient manual checking by doing both result and statistical calculations. In MVM, all calculations, results, and statistical are structurally validated by Waters and performed within Empower. Calculations can be performed easily with a click of a button. With MVM performing all calculations, there is no need for data transfer to third-party software packages and hence reducing transcription errors and time spent cross-checking data. Next, let's look into how MVM can bring about gold standard compliance for your laboratory. When I was traveling for work, I often had to sift through many crumpled receipts at the end of the month and try to decipher where and when I took each ride and to meet which customer. Occasionally, to my horror, I will come across some receipts with faded numbers that I can't claim. Sometimes, I might even misplace receipts. With Grab, I no longer had this worry. Information about all my rides could be compiled and exported, ensuring that I make my full travel claim every time easily. Similarly, with MVM, you no longer have to deal with misplaced reports of results or trying to sift through paperwork to search for information. Let's look at how MVM can give you data traceability automatically, giving you the convenience of data documentation and search, as well as allowing you to meet regulatory compliance. Since MVM runs on Empower, it contains the same regulatory and compliant-ready benefits. All data is stored in the embedded Oracle relational database, which provides security and data integrity. With Empower performing all calculations, validation results can be easily traceable to its chromatographic results, raw data, methods, and all metadata automatically. This allows for easy interrogation of your validation results. To investigate if there could be an outlier, just look into the raw data files in Empower. To check if the column temperature has been set correctly, you can look at the instrument method in Empower. To verify which system was used and if it has been maintained, you can trace the system information in Empower. Custom fields can also be used to enter information into Empower to aid in full traceability or for additional calculations. In this case, we can enter the product code as a custom field, so that it can be documented and subsequently searchable on Empower. Another use of custom fields is for the documentation of factors that are varied as part of a robustness study. With custom fields, a design of experiment where multiple factors are varied at the same time can be carried out with ease. In this example, the factors column temperature, flow rate, and detector wavelengths were investigated. The lower, upper, and nominal value that you see here are all documented with custom fields. Using a full factorial experimental design and with the help of the sample set generator, a robustness study sample set with different instrument methods could be automatically generated to investigate the combination of different instrument conditions. Finally, MVM allows for enhanced data security, allowing method validation requirements to be approved and logged at various steps in the workflow, reflecting your method validation protocol. This can be done at sample set method, sample set for raw data, result set for process data, validation result processing for process validation data, and finally, the overall validation study. Once the method validation requirements are logged, no changes can be made unless a user unlocks it. Are you now convinced that MVM can make a difference to your method validation workflow? Let's hear from a scientist and MVM user from a global pharmaceutical company R&D division who noted the benefits of MVM in this code. Non-sample prep activities account for approximately 60% of the time consumed in a validation study. With MVM, he estimates this could be reduced to as little as 10%, representing a 50% overall time savings. He continued to add that he is also more confident that his method validation data is accurate and traceable. Are you ready to take advantage of these benefits? Achieving peak efficiency, reducing risk, and having gold standard compliance. Please reach out to your Waters account manager for more information. For more information, please view our introductory video on MVM. This can be found on episode 10 of the Empower vlog. There are Method Validation Manager collaterals available for download via the resource list on this webinar page, and that includes the brochure, business solution, white paper, and application note. Finally, additional information on MVM can also be found on the Empower Tip of the Week blog by Neil Lander, and MVM is featured from tip 187 to 199. If you have any questions or would like to connect, please reach out to me via the QR code here for my LinkedIn or through my email address. Thank you for your time and hope you enjoy the rest of the LC Symposium.
We have come to the end of our session, reducing our specs results in Pharma Lab. Today, we have heard from a strong lineup of speakers on various tools and best practices to drive data quality and reduce OS results. By taking a proactive approach, Pauline described the use of risk assessments can increase your understanding of the parameters that affect your method and define control strategy to reduce variability when switching your method to a different system. Paula then gave us an excellent example of putting this concept in practice with successful method transfer to 4 ARC HPLC in labs across the globe. Choosing quality tool goes beyond just the LC system. Today we show how consumables and the subtle yet impactful changes brought about by our latest premier technology can help to ensure accurate analysis and reduce OOS when running method routinely. Lastly, not to forget the power of automations. Andrew Pass robot can relieve time-intensive and repetitive manual tasks that are prone to human error in sample prep, as much as Empower MVM software can on streamlining your method validation workflow. As a reminder, you'll be able to access today's recordings immediately after this live event, and you may submit your questions on Slido until the end of tomorrow, May 19. We will consolidate all questions and answers by our speakers as a post-event email follow-up. When submitting your question on Slido, please also participate in the poll question we have prepared for today's event. Thank you very much for your attention, and we at Waters hope you have found the pharma session enjoyable and informative. Have a good rest of the day. Bye.